Welcome to Science for Life. Today in this video, we discuss about a practical which are based on homology sequence search uh, by using a BLAST search tool. So in this practical, we discuss how to search a homology related to a particular protein on the database, and just like as the NCBI or any data bank. This practical is continue part of the earlier practical which are we taken in last video. The last practical is to retrieve a sequence of interested protein or any molecule from a net or NCBI. So continue with that. We taking this another practical that same protein which we take keratin. So that keratin protein and its homology sequences which are found on this data bank that we're going to search by using a BLAST search tool. So the BLAST is a basic local search alignment tool, which also one part of a NCBI. So we are going to search this homology sequences. So let's start to our practical. So first of all, this half part of this practical is the same as first practical, in which we're going to retrieve the sequence of that keratin protein, and the next part is homology searching. So both both practicals are covered up in this video. So let's start. For to retrieve or homology, for any purpose you required your one not PC or a mobile or any laptop in which you have a net connection, and this some sites like Google or any browser from where you going to search. Then when you open your Chrome browser, then fill it in NCBI. NCBI means National Center for Biotechnology Information. When you open this site, it will take you on the official homepage. When you go at the NCBI's official homepage, then there will be so many features are shown. So see at this left side, here is so many like a chemical bioassay, DNA and RNA, genetics and medicine, homology, literature, protein sequences, analysis, and so many. So these are the shortcut from where we search or choose any molecule which we are interested. If you don't want, then here is also one option like all databases. So here also we choose any wanted or interested way. So we are choosing as a protein of our interested molecule. Then after choosing this, we also there is one option search bar in which we have to input a particular protein name. So we are same inputting a name of a keratin protein which I take in last lecture, last practical. So input it and search it. Then after searching, it will take you on another page which show you a detailed information about that keratin protein. So how that protein and what contents it has. Yeah, now this page is open, and yeah, you are able to find so many things on this screen. So first of all, here is a species, and that all species are shows the animals which contribute near about 48,250 keratin proteins are present on this data bank or this NCBI. Then plants have 2,271. So just like a fungi, protists, bacteria, archaea, viruses, and so many others. So these are options, and below that there is a sources from where it take place. So it takes from a PDB. PDB means protein data bank, then REF sequence, then UniProt, and other options also available. Then there is another information about this keratin protein, and that is molecular weight and other things. So after searching, there is so many proteins are we able to see. This shows that near about 82,785 types of proteins, or that much proteins are shown related to the keratin. So these are the list of these proteins, just like a keratin from Homo sapiens, then this keratin from um, Pseudomonas aeruginosa. So just like this, so many means 82,000 proteins are shown as a result on this page. So at right side, there is another information about this keratin. That is top organisms which has this keratin protein. They are near about 9,377 in Serratia marcescens, then Epidermidibacterium and Carotheniaceae, then Bacillus and Homo sapiens. And there are so many animals or taxons which has related this keratin protein and their amount. Then another option is database. So here also that related data we easily search on this platform. So when you have chosen a particular protein, then there is accession number is must. So this accession number is help to find easily that protein and that protein's location in this data bank or NCBI gene bank database bank. So this number work as book when you search in a library your particular book. So that type this accession number is required. So we're going to copy it, then put it in our search taskbar. After opening a new window, you search it in a taskbar. After paste it, so you get a accession number like AB30058.2, then search it. So after searching you get the same result which are we are going in last slide. So it take you at that keratin protein, Homo sapiens keratin partial protein. So it shows you a detailed information like this. So in this here these are the whole sequence of the protein, the keratin protein, just like this from V to last is SS. These all are amino acids which are denoted by a letters of English, just like a B, T, L, A, R. Such type of letters are denoted as amino acids. So first of all, here is also a accession number, and then this is a FASTA. So FASTA and graphics is two options from by which we going to search their formats. There is one FASTA format which shows you in a text format, and graphics. Graphics shows you the graphical forms like a diagram or a chart. So these are the options, and then you see there is a origin of a source of organisms and Homo sapien, and it comes from a eukaryota, metazoa, chordata, and near about hominidae. This all description related to the evolution. Then references, journal name, and where it published, when it published, and the authors. That detail of that particular protein is shown in this information. Then at last, there it shows you the amount of amino acid, and that is 245 amino acids are present in this keratin protein. It has a four complete line and four amino acids. So this upper line is contain near about 60 amino acids, and whole are 245. So this is the actual sequence of a keratin protein. Okay, then here there is a send to option. So from this send to, we easily download by going a file and then create file. But before that you have to change the format as a FASTA format and then click on a create file. After that you see here is a your file is going to download. After the download you go and open that sequence of protein which are into a text form. So that sequence is also we able to see by the FASTA format because it's also available in a text forms. So this downloaded text we're going to see now. So there is a another way of downloading it to go by accession number and put it in a search bar. Then when you get the particular yeah, just like this, yeah, it will open. Yeah, in WordPad you copy it and paste it. So this is the excision number of a keratin partial protein. Yeah, and it from a Homo sapien, and these remaining are all is a amino acids which are denoted by a capital letters. So this whole are 245 amino acids present in this keratin protein which are taken from a Homo sapiens. So this sequence you have to keep for your further search. Then there is the option of a FASTA. So when you go and search as a FASTA, then it also directly show you as a sequence search. So this I show you this, copy it and paste in WordPad. This is another way, but you already download it, so it will use us for our next further work. So here is another option, that is run blasta identify console. Okay, so for here, the first practical after downloading or successfully retrieving our amino acid keratin protein sequence. So first practical is complete here, but of retrieval of a data sequence. But we have to go in to search a homology of that keratin protein. That next practical is start from here, actually from here, because we already have the sequence of a protein, that keratin. So we know that actual sequence. So we now going to search the related sequences or same sequences which are present on this NCBI site. So the next procedure, it quite a different from that. So let's go for the another practical, that is homology searches by using a blast. So you are able to see here, here is so many options at right side. There is a run blast, identify, consult, find, and articles about this protein also. And when you want to download it, then there is option now at near about every page. So let's go to our graphics. Yeah, first of all you have to go at graphics, how this protein in a graphic form looks like. So it shows you a graph form in a colored lines. Actually this line shows information about that particular protein which are keratin protein. So yeah, like this, these are a detailed information but in a form of a graphs. So yes, where you are going to click, then it shows you another information about the particular part or a particular sequence of that protein. And just like see here, here is a protein features, then region features. Yeah, just like this. So it's also easy or interesting to search it. But we have to go for a homology search. So we have to click on the run blast. Yeah. um, After hit the button run blast, it will take you on another site or another window where actual that keratin protein is compared with the um, other sequences which are related to that keratin which present on this NCBI site. So here in this window we see there is a interaction number. So already this accession number is related to our keratin protein, and yeah, this is related to our original keratin protein, which are our interested part or sequence. Then here is also options which shows you to find another one or another particular sequences. So for that you have to choose your directly. Here is option to choose your file. If you don't select it, then you go from here to our desktop or your files and select this accession number or directly sequence of that protein or any molecule. After that there is another options which shows you uh, databases from where non-redundant protein sequence or others. Then there is organism, from which organism you going to take that sequence of their common name and detailed information. Yeah. After this, yeah, this is a direct sequence which we downloaded that will copy and paste here. Yeah, that also a direct way to input your sequence or accession number. Yeah, after this, there is a algorithm on which algorithm that will work. Yeah, this is a quick blast or blast P, PSI blast or PHI blast. But we are yeah, perfect with the blast P protein. Okay, then there is a one option as a blast. So hit that button, blast. It means it will work on that aligning with our protein, with our similar proteins which are stored in this bank. So in a few seconds, it will show you a result. So yeah, see here. This compare whole data which in a form of proteins or amino acids, and that will analyze or compare and most related sequence that will match, and results will be shown very quickly. So see here, there is a program by which blast we search it, then database or citations. It will show you a detailed information as a references and from where we take it, then query ID, then description of where we keratin protein we take, then there is a query length, it means amino acid length 245. Then this is the actual result. Yeah, that is near about 100 result is shown in this list. So these all are the similar or homology proteins which are uh, present in this particular NCBI database bank. So yeah, you see here there is a max score. The first is our interested protein keratin Homo sapiens. It is 100% match with our sequence because it is our actual protein. Then you see the next protein, that is Homo sapiens, but it is related to the type 1 keratin 16 Homo sapiens. It's quite a different from us. Yeah, see here. So it is a 98.36 yeah, similar. So it's not actual 100% because some similarity is not there. Yeah, some difference is there. in amino acid sequences. So it shows a variation. So like that, this whole amino acids are near about same, but not exactly equal. So list is a 91, 86, and 85. And these are some similarities. But actually, here is some another parameters by which homology is done, like a max score, total score, query cover, e-value, per identity, X identity, and accession. So here is accession number is also provided of each and every protein sequences. So directly you also choose any accession number and go on a particular protein to get a detailed information about that protein, or directly you copy that accession number and furtherly use it. Yeah, just like this we select some proteins for our further study. uh, just here. Yeah, keratin. This is another protein. By selecting a direct accession number or clicking on it, we go on a direct this keratin type 1 cytoskeleton 16 protein, which are not exact similar but near about our protein. It's also provided all information, detailed information about that protein. But this protein is it's bigger than our keratin because it has a 476 amino acids, so it is a quite different. So let it be. We here is also option of downloading it, but there is not a requirement of our that particular protein. So we go back on our page and now select some yeah, now we select some proteins, but before that we choose some 2 3 proteins or 10 proteins, and just like upper 2, middle 2 or 3 proteins, and at a lower here is a 3 proteins. Yeah, like this, and one more. Yeah, here. Yeah, now these are 9 10 10 proteins we choose, and then now we're going to compare it with our actual keratin protein. Yeah, by using a graphic summary, or yeah, this shows you a similarity or homology between these 10 proteins. The uppermost is our actual keratin protein. Yes, keratin from Homo sapien. Then another is a 16 Homo sapien protein, keratin 16. Then this is a type 1. Then another is a hypothetical protein. Then next is a keratin type 1 cytoskeleton 16. So these are the rank, or according to their homology, how much they are similar to our original keratin protein. So and they are sequenced like this. Okay, and this lower is quite similar because we take it from a lowest, yeah, 100 number of on this list. So these are not too much similar with our original protein. Yeah, okay, here you see that these colors from black to red, it means when there is a black, then similarity is not equal, and when there is a red, then there is a, it is near about similar. So next we see these proteins, 10 proteins in alignments that filter. After alignment, that shows you a result in different ways. You see the actual in the form of a text. You see there, there is a text formats of actual protein of keratin protein that will match with another proteins. So this is a cytoskeleton protein type 1. This is a hypothetical. This is a keratin type 1, and this our homo sapien partial keratin protein. So it will match with our each and every sequence, and it show you a the percentage or amount of homology of that particular protein. Okay, then It also provide you a detailed information about that particular proteins, which are how much similar with our protein or not. Then when we go with the next format, that is taxonomy. After in a taxonomy, we compare the 10 proteins with our keratin, actual keratin. Then it shows these results. It shows you the organism's name in from which we take these proteins, and the blast name from where it is placental or primates or bats or any others, and the score how much their proteins are matched 482, 426, or how many these are, and a number of hits means how much change in these proteins are shown here. Then these are homo sapien hits. Yeah, these are related shows in a taxonomy. In taxonomy there is also one organisms. Yeah, it provides a particular organism's name and detailed information about that particular protein in which organism. So this also show you a score, e value, and accession number. So which protein is so much matched with us, we are easily get their accession number and the detail information about that protein by clicking on this or copying this accession number. Then next is a taxonomy. So after searching a taxonomy, we get the same result as shown in this upper taxonomy option. So here is a numbers of hits and number of organisms which are get a similar protein found in them. So like this root or macaca, then this myobis myotus, and there is a homo sapien hits, macaca mulatus hits. So these are organisms. Yeah, this is a detailed information about this keratin protein, which shows us homology of that tools, homology of another proteins which are similar to this. So then, after seeing this, uh, well, there is no any barrier to how much you compare the proteins. So we take only a 10 proteins, but there is so many options to compare it. And here is the option of a downloading. So we are download from any format. So we choose a FASTA format. So FASTA format shows us a text format. So it is easily get us the understanding of that sequences which are formed in a faster form or text forms. So it is quite easy to us. Then yeah, when you yes, select a download option, then after downloading there is a file is downloaded here. Yeah, first of all, I going to tell you that here is a download option. When you click on this download, then it shows you a format which FASTA or any other format. When you download it in a FASTA format, then download it here. And when you open this file or downloaded sequences files, which shows you like this. Yeah, this is actual sequence of that keratin proteins, but not only a exact one protein, it is of a 10 proteins which we select in that list. So there is a easy to sort out them, as where you see this greater than sign, it means from here the actual proteins start. So like this we see here is also another one greater than sign. From here we going to, yeah, from here we're going to splice it, yeah, split it, and it is a separate amino acid for sort outing. So like this we sort out a 10 amino acids step by step, and after finally sort outing each and every sequence, we get the exact this 10 amino acid sequences, and each and every one is particularly separate, or not exactly separate, but much similar to each and every one, because this last one, it is a very much differ than our original keratin protein. So like this, we successfully retrieve the proteins which are homology shown, or they are similar with our keratin protein. So I think here also option offer to download this graphical form also. So this graph also we easily download by the option, and use it for further research. So this type we compare our sequence with another proteins which are present on this data bank. So thank you.